Can anybody?
Yeah, we can hear you.
Okay, and I think we can, well, we can put up, whenever you're ready, you can put your camera on.
Oh, is it not on?
Yeah,
Oh,
not
yeah,
yet.
yeah. Here we go.
There it is.
There we go.
Okay. Hi, I hope you can hear us. So we're here with Deanna Barch today, who's here at UCSD for a couple of days. She's visiting the Department of Psychiatry, and as many of you know, but maybe not all of you know, she's a professor of psychological and brain sciences and psychiatry and radiology at Washington University in St. Louis. Deanna is the Vice Dean of Research in the Arts and Sciences at WashU and the Gregory B. Couch Professor of Psychiatry as well. And Deanna has done many decades of research that characterize cognitive functioning, brain connectivity, and mechanisms that subserve conditions like schizophrenia, depression, PTSD, and her research uses different psychological techniques, a lot of neuroimaging, and various computational approaches that span the lifespan from children through adulthood through later stages of life. Deanna does 1 million things. It does each thing incredibly well. Nobody can understand how it all happened. She also is the deputy editor at Biological Psychiatry, editor-in-chief of Biological Psychiatry Global Open Science. She's on scientific boards of BBRF, One Mind Foundation, the Stanley Foundation, and the Bipolar Discovery Initiative. Deanna was on the executive committee of the Association for Psychological Sciences and the Scientific Council of the National Institute of Mental Health, and she's president-elect for Flux and ACNP. Okay. I can go on. I'm done introducing her.
Anything else?
She's fellow for APS, for ACNP, member of the National Academy of Medicine and the American Academy of Arts and Sciences. Deanna is also a key leader in the adolescent brain cognitive development study. She is a leader of the site at Washington University and leads our mental health work group, which is an incredibly important part of ABCD. So I could go on and on, but I'd like to introduce you to Deanna so we can hear her talk about her science.
Thank you, Susan. Thank you. And thank you everybody for having me. You forgot to mention my karaoke skills, which are the most important thing, right? Yeah. So I thought I would do something a teeny bit different because it's kind of interesting. Like I know a bunch of you and I know some of you from different parts of my research life, right? And so a lot of you I know from ABCD, but Eric I know from more of my chronic mental illness world and Jared from my sort of translational world. And when Susan said, what do you want to talk about? I thought about it and it was, trying to think about like how to talk about my overall program of research. So rather than try to talk about too many things incoherently, what I thought I would do is just tell you a little bit about upfront about sort of how I think about my program of research, and then focus in on one particular line of research and sort of tell a story about that line of research. And okay, here we go. There we go. So don't have any conflicts to disclose. So thinking about my overall program of research, I think of the connecting themes as work trying to understand the neural and cognitive contributions to a range of different mental health challenges across the lifespan. And within that, I have sort of different threads of research that sort of tackle that from different perspectives. So one thread is focusing on the cognitive and neural contributions to depression. And the line there starts early because actually the very first project I did as an undergraduate honors thesis was looking at cognitive dysfunction and depression. And that is something that has actually kind of continued throughout my research program, going everywhere from looking at prenatal, preschool onset depression through to collaborations looking at later life depression and geriatric depression. I also have for a long time done work looking at cognitive and brain contributions to psychosis with a particular focus on kind of motivation and negative symptoms. And that is work that I've done partly in collaboration with Jared in various contexts. but really kind of trying to understand, particularly in people who have more chronic mental illness, what are the neural and psychological mechanisms that might contribute to cognitive dysfunction? How does that relate to functional impairment? How does that relate to negative symptoms? I've also been fairly involved in work just to try to understand typical brain structure and function, which I really see as integral to understanding atypicality, right? Because it's pretty challenging for us to understand what isn't going awry without understanding kind of how things are happening normatively. And that includes involvement in the Human Connectome projects. I was one of the PIs of the developmental extension of the Human Connectome, and that kind of really led to my involvement in the adolescent brain and cognitive development study. And WashU is now also one of the sites for the HBCD, the I can never get the acronyms right, Healthy Brain Cognitive Development Study, starting at that kind of very early end of life. And I mentioned negative symptoms, but I do think of anhedonia and amotivation transdiagnostically as sort of a key component. Because of my interest in both depression and psychosis, amotivation is something that characterizes both. It can also characterize people with bipolar disorder at different phases of their illness. And particularly with the advent of the research diagnostic criteria, there was a lot of interest in understanding what is transdiagnostic, what is not transdiagnostic, how do we characterize these, do they have the same or different neural contributions? I could give a whole separate talk about our work in that domain, where I will say we don't think it's transdiagnostic. We think that there are really different things going on in depression and psychosis, both cognitively, emotionally, and neurally. But we've still got work to tease that apart. Today, I'm going to talk about perhaps the kind of most recent thread of my research, which came out of work in the ABCD, but also work that I've been doing collaboratively. I'll talk about this in a little bit more detail with a team of people at WashU, where we started a study a number of years ago, 22 years ago now, where we actually followed kids longitudinally for 22 years. And we have a lot of data on early poverty and the impact or the relationship to long-term outcomes in these kids. And then I'll talk about some more recent work that we started actually in utero to understand sort of how early some of these impacts were happening, how that related to brain development early in life, and how that links to very early signs and symptoms of potential mental health challenges, even in preschool. So hopefully you see there's a theme there, right? Brain and cognition across the lifespan. And I think one of the things I've really learned over the years is that these different threads are mutually informative, right? And that there's some real benefits to not focusing only on sort of a single domain of mental health challenges, because I feel like you can get a pretty narrow view of what's going on, and maybe the illusion of specificity that changes when you start to think about many different forms of mental health challenges, particularly serious mental illness, and what might be shared and what might be distinct. And I always like to start by acknowledging my collaborators for work that I'm going to talk about, because I feel like often you run out of time and they get short shrift. So in particular, I want to thank my collaborator, Joan Luby, who's a child psychiatrist, and we have worked together on a lot of the developmental work that we're doing. And we've had this really fabulous group of graduate students, postdocs, early career faculty, a number of who come from the T32 that Joan and I co-direct on developmental psychopathology and neuroscience. And we kind of have a collaborative team of people who work together. And one of the reasons I chose to talk about sort of this line of research is I think it's also been a really interesting line of research in terms of the collaborations across disciplines that it's required to do this well. The work I'm going to talk about really needed psychology, psychiatry, radiology, immunology, neurology, pediatrics, OBGYN, and in the very end, I'll talk about economics and the important role of economics. And, I think that has been a really interesting experience, learning how to work with people with very different backgrounds and to kind of think about shared interests and ideas, but different sort of research trajectories and histories and experiences. So that's why I kind of chose to focus on this. And at any time, if people have questions, please feel free to stop me. I'm happy to stop and explain anything that's not clear. So to kind of dive into the work on early development, I think we all know that the causes of mental illness are multifactorial, right? Like we've known for years that genetics make an important contribution to almost every form of mental illness. There are also what I'll call individual factors that we have not really identified yet that are clearly important contributors. And we've known for a really long time that the environment plays an important role, right? So none of this is kind of new knowledge. In terms of the environment, I think historically we focused a lot, rightfully so, on abuse, trauma, life events, because we know those things are associated with an increased risk for mental health challenges. More recently, I think people have also started to focus on sort of what I think of as trauma at a more societal level, right? Discrimination, structural adversity, the ways in which that impacts and interacts with abuse and trauma and life events. But also childhood poverty and what I'll call neighborhood adversity, sort of adversity at the level of neighborhoods in terms of economic resources, safety, and other things, are elements of the environment that we now recognize um, have their additional contribution to risk for mental health. They certainly interact with some of these other things, but they may themselves also be potential contributors to risk for a wide variety of forms of mental health challenges. And that's where our work in this domain has focused, is really trying to understand what are the relationships between early childhood poverty and neighborhood adversity, and then what are the mechanisms by which that gets into the brain and into the body and sort of puts people at risk for mental health challenges later in life? And when I say mechanisms, we know that there's, by itself, it's not so useful to say, okay, childhood poverty or neighborhood adversity puts people at risk. What is it about that, right? So certainly we know that childhood poverty is associated with experiencing chronic stress and instability in the home environment. We know, unfortunately, that families that are struggling financially, it can lead to disruptions in caregiver support. And I don't mean that in a victim blaming way, but anybody who's ever had to work multiple jobs or shift work, try to deal with childcare issues, it makes it much more difficult. We also know increasingly that, living in neighborhoods because of sort of structural adversity and racism, often there's exposure to environmental toxins. We all know about lead, that's the common one, but there's many other toxins and other things in more impoverished neighborhoods, polycyclic hydrocarbons, other things that kids are exposed to that can have an impact on brain development. And, you know, I think there's a lot of evidence pointing to all of these things impinging on brain development in key ways that then put kids at risk for a variety of what I'll call brain health challenges. And I use a bit broader term rather than just mental health challenges, because I think it also often puts kids at risk for challenges in learning and cognition, emotion regulation, as well as mental health challenges. So I'm going to unpack some of the research that we've done that was designed to kind of look at this pathway and to try to understand mechanisms. And when I mentioned this study, long-running study, this was a study that I actually didn't start. It was started in 2002 by Joan Luby, child psychiatrist, who was really interested in preschool onset depression. And she was seeing kids in her clinic. She was a infant and early childhood psychiatrist. She was seeing kids that came in with what she thought was depression. And she was being told that can't possibly be true because little kids can't be depressed, right? Kind of came out of more psychoanalytic framework saying kids don't have the sort of ego development to be depressed. So she started a study where she started bringing them in and doing clinical interviews and parent-child interactions and measuring things about the environment and, emotion, function, cognition. And then right around when they hit school age, she came knocking on my door and said, we really need to study the brain in these kids. Can you please come? Can I get you excited about kids? Right. So this was my first foray into non-adult human imaging. And I said, sure, what the heck, let's do it. And that has led to this longstanding collaboration where we first had a supplement funded to her grant to do our first wave of imaging. Then we got several R01s to continue to follow these kids. We actually now have been refunded to follow these kids who are now adults into adulthood. not with imaging, but to really understand their life function, and with a particular focus, interestingly, on borderline symptomatology. We could talk about that. But we basically ended up doing, over time, 5 waves of neuroimaging with these kids. And we have, many, many, many different measures of assessments. But in the first study, when we started to look at this, what we wanted to do was look at the measures of poverty that she had obtained from preschool. And she had videotaped, observed caretaker-child interaction measures that had been coded. And we had life events and other sorts of things. And when we did this first project, there was a lot of really interesting animal research that was coming out from some really elegant models of like maternal care disruption, maternal bedding restriction, that were meant to sort of emulate early environmental deprivation and what impact that had on dams and their behavior towards their pups, and what impact that was having on pup development. And a lot of that work was pointing to it leading to disruptions in hippocampal structure and function, which was interesting from the perspective of the role of the hippocampus in sort of modulating responses of the HPA axis to stress and learning and memory. And so there was this really elegant work coming out in animals that at a very kind of molecular level was saying, okay, what's happening in the hippocampus and to some extent the amygdala in these pups when their moms didn't have enough resources, and then caretaking became disorganized. It had long-lasting effects on these pups. It even was transmitted to their behavior towards their own pups. So we said, well, what do we see any hints of a relationship like that in human children? And so what we did was focus on, in this first study, looking at hippocampal structure in MRI at the very first imaging way we did with these kids when they were 8 to 10 years old, right? So what did we see? Well, perhaps now, not surprisingly, but at the time it was a little more surprising, is we definitely saw a relationship between preschool poverty and hippocampal volume, such that those kids who were raised in environments that had less economic resources, when they were at school age, they had hippocampal volumes that were smaller, right? Hippocampal volume's a poor substitute for the elegant work that you can do in animal models, but it's sort of what we can do in human children at that age. Also not so surprising, greater preschool poverty was associated with children experiencing more stressful life events. And it was also associated with disruptions in the observed parenting measures that we had. So we saw less supportive and more quote unquote hostile parenting in families who had less economic resources. What was maybe more novel and interesting at the time was that both of these were also associated with hippocampal volume in these kids, and they were significant mediators of the relationship between preschool poverty and hippocampal volume. So we really think it's, it's sort of not just poverty on its own, but it's the way in which poverty is influencing things like parenting, life events, and other factors as well. Now, if you look at the literature now on early economic adversity and brain development, it's a big literature now. It's a little mixed in some places, but the hippocampal findings are really quite consistent. Almost every study that has looked at the relationship between early childhood poverty and hippocampal volume in childhood sees this relationship. And just to argue for replication, right? I've had access to this fabulous data set, some of you might know a little bit about, right? The Adolescent Brain and Cognitive Development Study. I don't think I need to tell anybody here what this is about. Oh, okay, I won't. But needless to say, UCSD leads this wonderful study of 21 sites across the country that recruited youth and has done a lot of work imaging the brain across time in these kids, but also really getting lots of elegant measures of individual family income and poverty, neighborhood adversity, other measures of, kind of structural adversity. And so we have been able to replicate these findings in the ABCD study. This is one study led by a postdoc, David Weissman, who's now faculty down the road from you in California. And he was postdocing with Katie McLaughlin at Harvard, and we started a collaboration looking at a variety of sort of environmental factors in brain development. So here I'm showing you a measure of income to needs. Oh, it doesn't work on that, does it? Okay, we will use this instead. So income to needs is a measure of poverty. It just indexes income in a family relative to the family size. So you can think of it as need, and it's thought to be a more sensitive measure of poverty. And then here I'm showing you internalizing problems at childhood. And what you see, as has been seen in many studies, is that lower SES, lower income to needs, is associated with children experiencing greater internalizing problems in kind of late childhood. And if you look at hippocampal volume again, you again see that lower SES, lower income to needs, is associated with reduced hippocampal volume, right? So this is a now pretty replicated finding. This focuses though just on family income, right? Which is super important. It's probably the biggest driver of those kinds of factors associated with income. But we know that families live in neighborhoods, right? And neighborhoods can have their own sort of um, experience of economic deprivation. And you might say, well, isn't it just the sum of families? Like, why would it be something different than just the sum of families? And often it is, but in many parts of the country, including my own home state, um, there's a dissociation because of structural racism. So I'll give Missouri as an example, has a long history of a variety of practices, redlining, covenants, other sorts of things that meant when Black families attained middle class income status and could afford, in theory, to move into a higher SES neighborhood, they were precluded from doing so. So you could have families that were living in neighborhoods, that had fairly, decent income levels, but the neighborhood as a whole might be more economically deprived because they really didn't have options to move out of those neighborhoods. So you can see some dissociation between individual family income and the whole neighborhood and sort of what they're experiencing. And fortunately, ABCD is a really great sample to look at that because there's variability across states and sites that sometimes gives you that variance that you need to understand those differential contributions. So we used a measure that a lot of people in the field use to look at neighborhood deprivation called the Area Deprivation Index based on census block measures. And it measures things like the median family income in a neighborhood, unemployment, people below the poverty line, a bunch of different indicators of sort of the average income in that neighborhood. And we looked at whether that continued to relate to child mental health concerns or brain development over and above individual family indicators of income. So family income, caretaker education, those sorts of factors. And we first looked at cognitive function, and indeed, even when controlling for these other family factors, greater neighborhood deprivation is associated with basically worse performance on every cognitive domain that we looked at: memory, executive function, reading, vocabulary skills, processing speed across the board. Now, these were not huge effect size, and ABCD is a very big study, so you can get very significant things that are not huge effect sizes. But, you know, in my mind, any impingement on kids' cognitive skills is not good for them. And then we looked at what might be mediating that in terms of brain structure, and we looked in particular at hippocampal volume and prefrontal cortex, controlling for whole brain volume. And what we saw was that both of these served as mediators between the relationship of neighborhood deprivation to these cognitive outcomes, but with some interesting specificity. So like hippocampus was more related to the memory measures, right? And the vocabulary measures, whereas the prefrontal cortex size was more related to the executive function measures, right? So again, some evidence that maybe by impinging on the brain, neighborhood factors are also impacting outcomes in kids. If you look also at mental health challenges, you really do tend to see neighborhood and family income related to most forms of mental health challenges in kids. But here's an example with externalizing symptoms. So this is from an analysis we did longitudinally in the ABCD data, where we were looking at trajectories of internalizing and externalizing symptoms over time in kids. And we said, are those patterns different in kids who are experiencing sort of low, medium, or high neighborhood deprivation? Again, once you control for family income. And so this is what the trajectory looks like in kids who have low neighborhood deprivation, so neighborhoods with higher SES on average, and then here's medium, and here's high. And what you can see is it's not really a slope change, right? The trajectory isn't so much different, but really from the beginning, those kids who are living in neighborhoods that have higher neighborhood deprivation are experiencing higher levels of externalizing symptoms, even again, controlling for family income, right? So there's something about the neighborhood. So, we were seeing these relationships, starting in school age. This is, kind of early adolescence. We've also been interested in how long these relationships last. That's actually a challenging question to ask in the US in particular, because we have now relatively little social mobility. So to really answer this question, really you need kids who say grew up in poverty and then were able to really move out significantly out of poverty and live significant parts of their life out of poverty. It doesn't happen very often anymore in the US, except under some unusual circumstances. Actually, graduate students are that unusual circumstance to some extent. Like, they could be related as kids being poor when their parents are in graduate school, but it's probably not quite the same thing as, families who are really living, with economic challenges. And there are hints from the physical health literature that early childhood exposure has sort of long lasting effects, where they've looked in the domains of like heart care, diabetes, other sorts of things. People who really significantly moved up economically, they continue to see greater, health, heart problems, greater risk of diabetes if they had lower income in childhood, even if they've been living for years in a high SES, lifestyle. So we were interested in, could we see any hints about how long this lasts? So we went back to the study with Joan, where I mentioned that we've been following these kids for a long time. Over time, we did 5 waves of imaging, and we did a follow-up assessment with them at the transition to adulthood, right? So they were like 18, 19, 20, and we were looking at cognitive outcomes at that age. Did they graduate from high school? Did they go into college? Did they develop effective social relationships? Were they engaging in high-risk behaviors? Did they continue to have depression problems? And what we saw was that those measures of childhood poverty were still predicting worse cognitive function, ongoing depression, greater high-risk behavior, worse educational outcomes, worse social relationships, even at that transition to adulthood. That was true even when we looked at SES in between, but in all honesty, there wasn't a lot of change in SES. So I don't think we ruled out that this isn't just ongoing exposure, right? But certainly the strongest relationships we saw were to the measures of income in the preschool era rather than like the adolescence era. Because we now also had access to these 5 waves of brain imaging, we put all that data together and looked at trajectories of brain development. And we did it at sort of different scales of granularity. So we looked at like whole brain gray and white matter volume. We looked at cortical versus subcortical gray matter volume. And then we looked at like particular subcortical structures. And what we saw was that pretty much across the board, we saw differences in brain structure associated with early poverty. And in some cases, these differences were differences in the trajectory of brain development over time. And when we controlled for everything and said, what is most strongly related to these outcomes? What we saw was that it was really subcortical gray matter volume that was most strongly related to these outcomes in kids. And if you looked at what was happening in terms of the trajectory, so here is a line, we did this as a continuous analysis, but just to illustrate the effects, I'm grouping them. So this is the kids with the lowest family income, and then sort of middle and highest family income. And there's 2 things that you should hopefully see in this graph. So the first is if you look over here, right, this is the sort of starting data that I showed you before, right? Reduced, I showed you hippocampal volume, but it actually isn't just hippocampal volume, it's broader across all of subcortex. But when you look at the trajectories, what you see actually is that the trajectory of growth of subcortical volumes is steeper in kids in higher income families, right? And it's shallower in kids in lower income families. And this trajectory, this slope over time, was the strongest mediator of the relationship between early poverty and these outcomes in kids over time, right? So we do think, again, that this early adversity is impacting on brain development in ways that are kind of predicting longer term outcomes in these kids. All right. We do see relationships to cortical volumes, but the subcortical are the strongest. So that, we got that data and we thought, okay, so we have to be thinking earlier in life. We have to be thinking about what interventions can we be doing earlier in life? Because at that point in time, my growing developmental hat was saying, like, the more we can help kids be on a typical developmental trajectory, have all the experiences that they need to develop effective emotion regulation, executive function skills, this is going to be protective against mental health challenges. So we got to really figure out how early this is starting so we know how early we need to start to intervene. So we started a new study that we call the Early Life Adversity and Biological Embedding Study, because everybody's got to have an acronym, right? So we call it ELAB. And this started a little bit before HBCD started, and kind of was kind of a precursor to WashU's contribution to the HBCD study. But we capitalized on sort of a unique opportunity we had where the March of Dimes had funded this huge study at WashU on moms and trying to predict prematurity. So they were recruiting all these moms and doing a bunch of measures on moms, including immune function, uh, 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 looking at circadian rhythms, melatonin, getting a bunch of interesting measures. And they were completely uninterested in the offspring. Like literally they refused to fund anything about the offspring. So we said, hey, can we recruit your moms, add some measures? So we added microbiome measures to look at the gut microbiome, and we added measures of like depression and stress and anxiety. And then we started imaging their babies at birth. And we have been following the babies and moms over time. We just got refunded to go to years 6 and 8. And we've been measuring tons of things, multiple ways of neuroimaging, although COVID happened right when the kids were 2 years old. So that was not great for our longitudinal neuroimaging. But we have inflammatory markers on moms and kids. We have gut microbiome on moms and kids. We have parent-child interactions. We have EEG. We have circadian and sleep measures on mom and kids. So really trying to hone in on like what's going on early on that might be, again, part of the mediating pathway. And then starting at about 1.5 years, we start to get sort of infant temperament and now early sort of dimensional precursors of mental health challenges in kids. And now that they're 6, we're doing starting to do full diagnostic interviews and look at sort of early emergence of child psychopathology. And this was the project where we have pediatricians, OBGYNs, immunologists all involved because it's a bunch of different things. So what do we see? So we focused on trying to think about what are all the things that might be impacting mom during pregnancy. We had 3 trimesters worth of data. We had a lot of different measures, and we weren't sure if everything was just going to be one big glom, like depression and stress and poverty, just one thing that's happening, or whether there are kind of dissociable factors. So we started out taking all of the measures that we had and doing a factor analysis and some structural equation modeling to say, is there kind of 1 factor, 2 factors here? And it turned out there were 2 dissociable factors, one that we're calling social disadvantage, that includes income to needs across all trimesters, our friendly area deprivation index, insurance status, education, and a little bit of diet, which is influenced by economic resources. And that's separated from a psychosocial stress factor, which was depression, experiences of racial discrimination and life events measured using the strain, and then mom's perceptions of distress. They were correlated about 0.35, right? But that actually wasn't as high as we thought they were going to be correlated. We kind of went into this thinking that these were going to be sort of 1 factor, but they're really pretty clearly dissociable factors. And the reason that's important is that there's been a lot of really good work on maternal depression during pregnancy and its impact. And a lot of that work has not measured SES and has not controlled for SES. And I do fear that some of the findings in the literature that are being attributed to maternal depression may actually not be maternal depression, but maybe instead mom's exposure to adversity during pregnancy, which, as you'll see, has a lot of impacts. So we got these measures, and then we use those measures to relate to brain outcomes at birth in these kids. So we imaged the kids between like a couple days. The latest kids were within like 28 days of birth, but we really tried to get as close as possible to the birth period so that there was less room for things to be happening to the kids post-birth before we did imaging. And so I'm going to show you, we've published a number of papers now on different metrics, but I'll show you the brain structure metrics as kind of the primary outcome. And so what I have plotted here is our disadvantage measure, which is that kind of factor score of those different things. And this is cortical gray matter volume at birth in these kids. And Chris Smizer, who leads the DAIC for the HBCD, is our kind of neonatal imaging guru on all these projects. And kind of sadly, what you see is that relationship is there at birth, right? And it's there if you control for gestational age, birth weight, NICU stay. If anybody knows Chris is like looking at every brain scan, like any little thing, control for all of that, some of which are also related to mom's experience of disadvantage, right? Birth weight and gestational age, but even controlling for that, we see reduced gray matter volumes. We also see same relationships to white matter and subcortical gray matter volumes. And we do not see any relationships to the psychosocial stress measures in mom, right? Once you control for SES, it's really all about the SES measures and not about maternal depression. In fact, we've struggled to find independent relationships of maternal depression to outcomes in this sample. We do see some relationships to parenting, but really all the variance is being sucked up by the sort of disadvantage measures here. We've also looked at diffusion measures, and we see something very similar. It's not every diffusion measure, right? So it's kind of more specific, but for example, in the cingulum, we do see that mean diffusivity is related to social disadvantage at birth. And we have a paper under review, hopefully almost accepted, that looked at functional connectivity at birth and also sees disruptions, particularly in kind of frontal parietal and somatomotor networks, as well as subcortical networks at birth. So whatever relationship is there, it's starting already during pregnancy. And you might say, well, what is happening during pregnancy that might be mediating it? And there's unfortunately a number of things that are happening. One of the big things is sleep disruption. So anybody who's been pregnant knows that there is normative sleep disruption happening during pregnancy, but over and above that, if you look in particular, not just at sleep duration, but what we call chronodisruption. So irregularity in measures of sleep, right? Predict over and above sleep, like just average sleep duration. So this is looking at what we call daily deviation in sleep duration. So it's looking at the variability over days. And that is strongly related to maternal social disadvantage. And it is a mediator of infant cortical gray matter volume, as well as infant cortical white matter volume. And quite frankly, we've now looked at outcomes at ages 2 and 3, and the maternal chronodisruption during pregnancy predicts cognitive function, and it predicts both, particularly externalizing or like early signs of I hate to call it externalizing a 2-year-old, attention sort of disruptive behavior disorder at age 2. And frankly, it's such a strong effect that it led us to write a Conti Center grant all about tackling chronodisruption, because there's been a lot of attention to sleep, but not much attention to chronodisruption, right? And these moms show chronodisruption in their sleep and in their melatonin secretion during pregnancy that seems to be be part of the pathway here. The other, so go here, I get ahead of myself. Okay, the other thing that we've been looking at is pro-inflammatory cytokines. We started looking in blood. We also have placentas. So we have a graduate student now who, for her dissertation, is going to look at placental cytokines and gene expression as a mediator. But the March of Dimes study collected circulating cytokines as part of their parent study and then shared them with us. And what you see in moms is that moms who had low SES, so we looked at a panel of IL-6, IL-8, IL-10, and TNF alpha. We would have loved to look at more, but it's pricey. And what we saw most strongly related was IL-6, which is a pro-inflammatory cytokine. So moms who had low SES had greater levels, circulating levels of IL-6. And then we looked at how those related to white matter development in their offspring. And we were interested in white matter in particular because of the potential role of inflammatory processes in disrupting white matter development. So first I'm going to show you, this is just the relationship between SES and different measures of white matter development, fractional anisotropy, mean diffusivity, and axial and radial diffusivity. Now, any of you who are white matter aficionados, if you haven't worked with neonatal data, all the relationships go in sort of the opposite direction of you would expect with the older kids, right? And that likely has to do with the nature of white matter development and contrast at that age. But what we see is that higher socioeconomic status during pregnancy is associated with greater mean diffusivity and axial diffusivity across a number of tracks, the cingulum bundle, looking at the uncinate fasciculus, a number of tracks show this relationship. And then we looked at the relationship to maternal IL-6, and particularly with axial diffusivity, you now see the opposite relationship, right? Where, again, SES is related to IL-6, and IL-6 is also related to white matter integrity in these kids, particularly axial diffusivity. And we now have year 2 and 3 data that we're looking at whether mom cytokines continue to predict at 2 and 3, but we now also have kids circulating cytokines and activated cytokines where when we take blood, we put it in a vial with lipopolysaccharide so you can get this activation of the cytokine measures. So we're going to look at whether that's kind of continuing on in the kids or not. So during pregnancy, mom's sleep, mom's, cytokine levels are associated with SES exposure, and those seem to be perhaps contributing mechanisms in utero to brain development that we're seeing already at birth. The design of the study, though, is right. We had birth measures, pregnancy measures at birth, and then we have all these measures now during the first couple of years of life, right? Where unfortunately most moms who were experiencing economic adversity during pregnancy are continuing to experience economic adversity because, again, we don't have a lot of social mobility. So we've also been looking at first year of life factors that we think might be also influencing brain development in kids. And we've been looking at what we've been calling the Thrive factor, which is looking at nutrition during the first year of life, environmental stimulation, caregiving, sleep in the kid. We now are actually getting polysomnography measures in the kids, and we'll be getting Fitbits, but in this case, this was just a parent reported measure, right? So not a great measure of sleep, but a starting hint. And then neighborhood safety, because we've been interested in sort of kids' experience of safeties. And you can kind of put these together into a factor that we're calling the Thrive factor, that we're kind of thinking of these are the things that kids need to thrive, right? Like they need caretaking, they need good food, they need adequate nutrition, they need to sleep, and they need to be safe, and they need to have stimulation to kind of be thriving during the first couple of years of life. And we're looking at those in relationship to outcomes now at year 3. And what we see is that that Thrive factor mediates the relationship between prenatal social disadvantage and both cognition at year 3 in these kids and gray matter volume at year 3 in these kids, right? So, moms who experience more economic adversity, right, have more challenges with nutrition and sleep and safety for their kids and parenting. And that is relating then to the kids' outcomes at year 3. And we've just done some more recent analyses to look at, again, early symptoms. And we're also seeing that this Thrive factor relates to internalizing and externalizing symptoms. We think some of these things are potentially actionable. I don't say that lightly because I know, it's easy for us to say, oh, you should just have your kids sleep better and get better food. Like that is not easy to do if you are struggling financially. But these are potentially mediating factors that we could think about interventions. And there have been interventions done with some of these things that show positive outcomes that are not universally used, right? So we've been starting to think about sort of clinical interventions that could be operating in the early years of life that we could be looking at very early signs and symptoms of mental health as outcomes. The other thing we've been looking at is kids' gut microbiome, partly because WashU is a big gut microbiome place, and that kind of got us interested. But if you're thinking about inflammatory pathways, the microbiome is actually a really important part of that, because part of what can be happening is diet and other factors can be changing the microbial composition of the gut microbiome. So this is data from 4 months in the kids. So we got these poop kits where moms would send us their poop, and then they also send us the diapers from kids, and you can analyze that data. And so here, what I'm showing you, and you're going to have to kind of trust me, but if you look at what microbes are more populating the gut microbiome of kids whose moms experience low disadvantage versus those who experienced high disadvantage. And if you look at the ones that are more populous in the low disadvantage kid, they tend to be more anti-inflammatory microbes, and the ones that are populating more frequently in the high disadvantage kids are more pro-inflammatory microbes, right? So now we're looking at, to what extent does this gut microbiome in the kids also relate to outcomes at years 2 and 3, right? And those analyses are ongoing. We haven't done those yet. All right. Okay. Am I out of time, or do I have any more time? You tell me.
We'll take a little time for questions.
Okay. Well, I'll end on one kind of study that brings in the economics piece, right? Because we've been trying to think a lot about, okay, we've identified all these relationships. What do we do about this, right? And I mentioned thinking about interventions, and that is something we're actively talking about. ARPA-H, which is a new funding mechanism, just came out with a new call that's really targeting sort of early life. And this is a domain where they would like to fund more things that would be good. So that's a possibility for us. But we've also been looking at public policy and trying to understand how public policy might mitigate some of this, because through the ABCD, NIDA has asked us multiple times to kind of come and talk to Congress about some of this work. And I think a lot of times people are like, well, what can we do about this, right? Like, and it's true, and you don't want to get like immobilized of like, this is an immutable thing that we can't do something about. So the same wonderful postdoc, David Weissman, decided that they were going to look at state level policy differences and understand whether or not that moderated the relationship between family poverty and kids' outcomes, right? With the idea that, well, first, this is one of the really great, you could never do this without the ABCD. Like, this is, to me, like one of the biggest things about the ABCD is you could never ask these questions without a countrywide study with multiple states that have different policies. And one of the things was that he noticed that the relationship between poverty and hippocampal volume and between poverty and internalizing problems in kids was not the same across every state, right? Could be noise, right? Could just be noise, but maybe it's meaningful. Maybe there's something different about those states that is leading to differences in the slopes of those lines, right? A stronger relationship versus a weaker relationship. So we saw it and thought about it, and we thought, well, there's multiple things that are different. So one big thing is the cost of living, right? So there are states with the higher cost of living. You'll notice where your state is in terms of cost of living. And then you'll notice where my state is, Missouri, in terms of cost of living. So certainly, right, the same income level means different things in different states, right?
See why you should move here?
I know, every time I look at this graph, I'm like, yeah, yeah.
Look at that graph more.
Yeah, but okay, I'm going to show you, there's other things that go along with that, right? So There's also the generosity of anti-poverty cash benefits that this is a state, and I actually didn't know this before we started this work. I think I had in my head that this was a national level policy, but it's not. Like the generosity of cash benefits are a state level policy, and states differ in how generous they are, right? So you have ones, again, look where your state is, right? Higher cash benefit versus lower cash benefit, right? And there's a variety of these benefits. This includes like food stamps and other things. So, whoop, let me go back here, right? And then there's also Medicare expansion, right? Which is another factor that's key, right? That some states have chosen to engage in Medicare expansion so that families have more access to healthcare and other states have not, right? So what happened? I must have lost a slide in here. So what he did was then an analysis where he looked at the interaction of these different factors, right? Is it a high or a low cost of living state? Do they have generous anti-poverty benefits or not? Do they have Medicare expansion or not? And what we looked at was, you know, kind of overall levels of symptoms and hippocampal volume, but also whether these things change the strength of the relationship between individual family income and outcomes, right? Because if you have buffering mechanisms, right, then the negative impact of low SES in a family is going to be less apparent because there are these societal factors, public policy factors that are mitigating those negative impacts, right? So what do you see? So I'm going to show you a bunch of slides that have sort of the same format. So first I'm going to show you the higher SES families. And the two bars on the left are the higher cost of living states, and the two bars on the right are the lower cost of living states. And then the different shading indicate higher, low cash benefit, right? And for higher SES families, it doesn't make a difference, and it shouldn't make a difference. The outcome here I'm showing you is hippocampal volume, right? Because those families have high income, these things are not going to impact them. But when you look at lower SES families, now you start to see a difference. So first, here are the higher cost of living states and the lower cost of living states. Across the board, hippocampal volume is lower for the lower income families. But what's critical is if you're looking at particularly higher cost of living states, right? Hippocampal volume on average in those states is different if it's a high cash benefit versus a low cash benefit state, right? And so it is particularly bad if you live in a high cost of living state that has low generosity of anti-poverty policies. And if you look at the slopes of the relationship between family income and hippocampal volume in those states, those are the steepest slopes. The strongest relationships between family income and hippocampal volume are in if you're in an expensive state that doesn't offer a lot in terms of anti-poverty programs. If you look at Medicare expansion, you see something similar, right? Doesn't really make a difference if you're a high SES family, but if you're a low SES family, again, the biggest effect is for high cost of living and no Medicaid expansion, which was my state for many years. This is hippocampal volume. You really see the exact same thing with internalizing symptoms. The graphs are flipped now, right? Because, you know, lower internalizing symptoms are better. So high SES families don't see a lot of difference. But if you look at lower SES families, overall, the kids have more internalizing symptoms. And that's particularly true if you live in a high cost of living state with low cash benefit. And if we look at Medicare, we see the exact same thing again, right? The highest internalizing symptoms at a mean level are the states that are expensive, right? Have lower SES families and don't have Medicare expansion. And again, if you look at the slopes of the relationships, the slopes are biggest in those low generosity, high cost of living states, right? Because they don't have those things to buffer and break those relationships. And I will say seeing this data made me change how I thought about, you know, if I'm going to talk about public policy interventions, I had been really thinking, oh, we got to talk to our Congress people and we got to talk to, you know, and there certainly are things we can be doing at a country level, but I think this really points to the importance of state level politics too, and being advocating at the state for what are the things that are going to be helpful to your state. And it was interesting. I was in Amsterdam last year. There's a woman named Eveline Kroon, who's a big developmental cognitive neuroscientist, and they have a big project in Amsterdam about growing up in society. And I presented some of this data, and I was saying, oh, well, you know, you can't really look at this in in the Netherlands because you know, you've got great social safety nets everywhere. And they said, no, that's actually not true. It's actually at a much more local level. And so we were talking about the fact that you could look like within the Netherlands, you could look at differences because there are very different policies across these much more granular measures. And for example, across countries in the European Union who have different policies and how that relationship might be impacting things. If they only had an ABCD study, we could look at this. So, all right, so I'll wrap up. So, you know, I'm pretty convinced that poverty gets under the skin and into the brain, and that it is a key risk factor, not deterministic, but a key risk factor for brain health challenges. And this has been a public health crisis for years. It's not like I need to tell you that, right? But like, I'm like, how much data do we have to show for us to take this more seriously? Um, I will say I could go on about the evidence for causality here, right? Animal models certainly help with that, but there are experiments of nature in humans that also really suggest a causal influence. And we have a paper that's just coming out, you know, looking at genetic, polygenic risk scores for educational attainment, all kinds of things. And you still see all of these poverty environmental effects, even when you control for those. Not a perfect study, right? Because we don't have the parent genetics, but you know, I think there's a lot of evidence that this is not just a, you know, sort of genetics are causing this, right? That there really is an environmental effect. And I think for me, this keeps pointing early intervention and, you know, for kids. I didn't show this because I thought I wouldn't have time, but we've also looked at this for psychosis risk, and we see these same environmental effects, and there's growing evidence about, you know, the impact of environmental adversity and poverty and discrimination on risk for the development of psychotic disorders. You know, so it really seems to be a pretty pervasive thing. And, you know, maybe this is just, you know, having been in the field for a while, I keep thinking like if there's something we can do that impacts many different mental health challenges, right? Like that would be a great thing to do, even if it's not specific to a particular mental health challenge. And I think a lot of the early adversity and environmental economics really are those kind of broad risk factors that are not specific, but in some ways you could flip that and say that's a positive thing because you have an opportunity to have a big, broad impact rather than, you know, kind of a focused impact later in life. And continuing to look at mechanisms, you know, there's a lot of things that seem to be going on: inflammation, sleep disruption, microbiome, caregiving. Some of these definitely seem like they could be targets for intervention early in life in ways that might be able to really help kids and families early in life. So I'll stop there. Sure, of course.
Question. I'm not quite sure I fully understand the difference between what you describe as neighborhood disadvantage and neighborhood adversity. But one of them you mentioned, but don't see any studies on, has to do with exposure to toxins. And when I think about how that can have such a pervasive effect, and we've already seen that families of lower socioeconomic status are not only exposed more, but have worse outcomes in terms of their neural development. I wondered if you could think about how that fits into this broader picture that you painted about the impact of neighborhood factors and just general exposure.
No, that's a good question. I mean, I think you could do a whole dismantling of neighborhood, right? Because I think there's multiple things. So I think one, when you think about communities and neighborhood, right? You know, if you live in a community that is relatively more economically advantaged, and your family's struggling, right? You have neighbors that you can go to for help, right? And social support and social buffering. So one piece of that is likely that if the whole community is economically challenged, right? You know, there's not a lot to go around, right? There is all the exposure to toxins, right? still lead. We still have a huge problem with lead exposure and lower income housing, but we have all these environmental toxins too, right? So the polycyclic hydrocarbons, because they tend to be closer to highways, right? Ambient particles in the air. We know that has an impact. The lack of green space probably has multiple impacts. There's probably also a piece of sort of the stress associated with neighborhood safety, right? So, and families will talk about that maybe they're afraid to let their kids out to play because there might be gun violence or other things going on. And that's assuming that there's a safe place for the kids to go play in a green space, which there often isn't. At least again in Missouri, what we see is that those neighborhoods tend to still not get access to the same, the kind of services that the government is supposed to, so their roads don't get fixed, the garbage doesn't get picked up, reliably. So we don't know what so I really think there are multiple facets there. I've had a bunch of grad students who were really interested in the neighborhood safety piece and have been trying to say is there a unique contribution of either perceptions of safety, like parent and child, ABCD has that, we have that in other studies, but also like indicate, like number of neighbor crimes in the neighborhood, right? Again, ABCD has that, and we have in our ELAB project, we had an MSTP student who worked with the geospatial people to get estimates of crimes and look at the impact of that. So I think that's as multifactorial, and it could be each of those is making a contribution. Yeah.
And just the, sort of one of the process features of how those fears about related to neighborhood safety impact, physical activity.
Yep.
and
That's another
which
good
then
point. Yep.
Impacts
Yep.
that variety of factors in
Yep.
growth as well.
Yeah. Yeah. If you don't have a place to go play, right? If you can't go for a run, you can't go for a walk, those kinds of things. It's a great point.
Definitely.
Yeah, a couple questions about this last really fascinating bunch of stuff that you showed us at the end there. Really specifically during pregnancy, implicating fetal effects.
Right.
So just you look, the relationship you're referring to is kind of flowing from SES kind of factor with cortical volume, which is a newer question that's not brain size.
It was thickness, I think. Yeah. Or well, no, it was cortical volume. Yeah.
So I'm assuming that's not brain size correct.
We've looked at it both ways, because Chris, makes it look at, but so it shows up in the global measures, even if you do brain size correction. And then if we're trying to do individual, like hippocampus or something, we'll always correct for the larger volume. We don't see a lot of evidence of specificity at that early age. It seems to be a broader brain thing, but it's over and above just like intracranial volume.
So, yeah, a couple of questions that I had. I mean, did you have substance use during pregnancy, and did that include antidepressants? So these effects all survive that. And the other thing is just, did you see it birth?
Yes, absolutely. So we have a first paper that we published with just the birth outcomes. So we looked at gestational age and birth weight. Both of those are also impacted, and it's related over and above. So the biggest substance use in our sample was alcohol and tobacco and cannabis. Not a lot of other substances, but we did have those too. And you can either leave those moms out of the analysis or you can control for them. You still see it. We have, antidepressant use. Cynthia Rogers, she's one of our collaborators, big interest in that. So all those effects survive that. And then when you do the brain stuff, we additionally control for birth weight and gestational age, and you still because I do think gestational age is a big part of this pathway. And this collaboration came about in part because Cynthia and Chris actually had started doing prematurity work, and then kind of brought me in when they were moving into younger kids, right? Because they were doing birth stuff. And so we were interested in the prematurity effects, and that's definitely part of it. Like that does suck up some of the variance, but there's something over and above the prematurity that's also related to the brain volumes at birth.
some kind of growth restrictions.
Yeah.
Above and beyond. what you see as such.
Yeah,
Yeah, that was
Sure.
incredibly elegant and very sophisticated work. Thank you. It's been a very an exceptionally clear presentation. I learned a lot. You had some interesting effect sizes. Some of those effect sizes, as we've seen a lot in literature, ability, relatively small,
particularly the ABCD stuff, yeah.
Be that as it may, still see some subgroups, and I guess that's my ultimate question. Are there subgroups? Are there factors that are additive that really put you at enormous risk that we should, from a policy perspective?
Yeah.
really
Yeah, so that kind of led us down the Thrive pathway, was saying, let's not look at each of these individual things, but let's think of like aggregate measures to kind of get at a bigger effect size. Because partly it's hard to dissociate these things. And when you do, and you're trying to say what's really the unique variance associated with it, that's a much smaller effect size. So we've been starting to, when we've been thinking about intervention development, thinking about a package of things that you could target simultaneously, with the idea that maybe that would have a bigger effect size. Because like the search for specificity gets you at smaller and smaller effect sizes, right? And the effect size for the overall relationship of SES to any of these outcomes is not small. It is by far the biggest effect, the number one biggest effect in the ABCD data set. We have a paper about that too, that it is the number one biggest effect. And it's a, 30 to 40% of the variance, right? Which is, no, it's not 70%, but it's pretty juicy. It's then when you start to say, well, what are all the mediating factors? Each of those accounts for some, and so the Thrive seems to be additive. Among the Thrive factors we've measured, parenting is a biggie, like that is a real biggie, as is the sleep. But there's a contribution of all of them, and so I do think it would be a mistake to say, okay, we're just going to go in and do parenting, or we're just going to go in and do sleep. I think it's a package that you have to look at together if you're really wanting to move the needle on the outcomes in the kids.
It's interesting, so if you were to actually do something definitively on a socioeconomic level, it would be generalized to the public. Just improving the overall SES as overall implications.
Yeah. And I think, so there's a study that we haven't been involved in, but is called Baby's First Years. But UCSD has been involved in that, right? And they are continuing to look at an income transfer model, right? So they had two groups of single moms early. One mom gets $333 a month, the other mom gets $20, and they're looking at outcomes on the kids. And no other intervention other than them seeing the people on a regular basis. So far, well, yeah, well, right. Yeah, well, they couldn't get the money from the government to do the income transfer. That had to come from the Gates Foundation because the NIH wouldn't do that, but they'll do the other parts of the study. I will say that the effects on moms really like being in the study in general, and there's an interesting paper about that. The effects on kid outcomes are modest so far. And Joan and I have talked about this a lot because we think that, one of the challenges is, you also have to think about intergenerational trauma and intergenerational transmission of things, right? Like money is important, although I would say $333 a month is probably not enough to really change your life in ways, but, I also think that people who have been systematically discriminated against, if you give them the resources, the evidence is that they will use those resources in good ways. There's economics folks have done this, but then they may not know how to access some of the things, right? So we're doing this little sub-analysis in our ELAB data set where we're looking at during COVID, who took advantage of the child. There was I forget what it was called, but there was this extra money you could get. And we thought, oh, surely it's the families that are struggling the most. They got access to this extra money and maybe it helped them. Well, it turned out it was the parents who had the most education who were most able to access that, right? So I think it's maybe initially not going to be enough just to kind of throw money at the problem, but we need to be thinking about, you know, like we think of it as like, you know, you can imagine like a resource helper who like you change the economics, but then you also start pointing to like, okay, and here are resources that you can use and how can you, you know, and sometimes people don't know, you need to educate them about the things that are helpful. So, you know, I do think I think it's a tractable problem, but it is a complex problem.
I hope the politicians can hear this.
Some of them can, some of them, others cannot.
Working on this, right?
Well, you know, there's all the, I just want to say it is interesting to talk to the economists because there's all kinds of long-term outcome data showing the long-term economic benefits at a state and country level to these things. But those are long-term and politicians are short-term because they have to get reelected, you know, and so it's just a very difficult problem because the people who need to vote on it are the people who need to see outcomes that their constituents care about now, not 15 years down the road.
Especially with 2-year terms.
But your model is, I think, remarkably consonant with this notion of precision medicine.
Yeah.
Because really, it's providing the needs for the individual based on, in this case, perhaps the type of disadvantage that
Right.
they're experiencing. And the precision part is helping to point that out
Yeah.
for them. What are the avenues that would be most
Yeah.
helpful for them?
And notably, I mean, this is something we've talked a lot about for ABCD, like we're pretty much looking at urban poverty, right? And rural poverty is actually the largest percentage of poverty in the US is rural, not urban poverty, which is a misunderstood thing about poverty. And I think some of the factors are similar. There's still greater exposure to toxins. It's just agricultural toxins now instead of other things, and lack of access to healthcare and all sorts of other things. But that may need a different set of interventions than urban, you know, economic disadvantage and outcomes. Yeah.
Well, thank you so much.
Thank
Deanna.
you. Thanks for listening. Oh, yeah, I can't.
No specific questions.
Great. All right. Thank you.
It's so nice to have you.
It's so fun to be here. It's so nice here.
Yes.